present in chili waste. So you have 15 minutes, please, Asma.
Yeah.
And I want to remind the participants that you can register in the chat for the certificate of attendance. So please don't forget to sign up. Go ahead, Asma.
Hello, everybody. First, you can see my slides, right?
Yeah.
Yes.
Okay. So I'm Asmaa Shakir, a grad student in Food Science and Technology from Department of Family and Consumer Science in the New Mexico State University. Today, I'm going to talk about my research identification and characterization of bioactive compound present in the chili waste. This study is focused on byproducts utilization from food industries by identifying this byproduct as a source of bioactive compounds with a potential industrial application. The objective of my research is identify, this is the specific objective, is identify and analyze the carotenoids compounds in the chili waste and identify the phenolic compounds and determine the antioxidant in the chili waste, then identify the total dietary fiber in the chili waste. Today, I'm gonna share with you the part of my study that involved for the recovery of the functional phenolic compounds from the chili waste. So the objective of this study is a quantitative, the total phenolic content in the chili waste, and then determine the antioxidant activity of the polyphenols in the chili waste. The phenolics are one of the major and diverse group of the active compounds in the plant. And chili pepper are a good source of the phenolic compounds with antioxidant properties. And phenolic compound can have the ability to destroy radicals. Also, phenolic compound influence the quality, acceptability, and stability of the food by acting as flavonoids, colorants, and antioxidants. Here we can see the total phenolic compounds in some vegetables and the fruit, and that to comparing this content with the chili. We can see, from example, for the broccoli, it's content 88, and this is measured by milligram by 100 gram of the sample. For tomato, for example, it's 68. For green tea is 83. And we can see here the higher concentration of the chili waste in the of the phenolic compound in the chili. So it's a higher concentration comparing with other vegetables and foods. Phenolic compounds are divided into different groups based on this chemical structure. Here we can see many structures of the groups of the phenolic compounds. We can see all of them contain aromatic ring with one or more hydroxyl group. So this is for example, this is the salicylic acid, which is common of the phenolics. This is stilbene. This is ligands, which is found in the seeds and whole wheat. Tannin, which is found in coffee and tea. A flavonoid, which is found in the hot peppers. It has been confirmed that consumption of the phenolics-rich food or beverage prevent some diseases. Here I summarize some the benefit of the phenolic compound. It is play important role in the cancer prevention by protecting DNA. It can reduce the insulin resistance and give a protection against diabetes. It also is it's beneficial in the improvement of the lipid profile and maintenance of the blood pressure. The antioxidant, as we know, is the substance that can prevent damage to the cell caused by the free radical, or we can say it can inhibit the oxidation. And free radicals are formed when we breathing and smoke or pollution, or sometimes exposure to the ultraviolet light from sun. So this can causes a higher amount of free radical in our body. Here in this slide, I can explain how the phenolic compounds can inhibit the free radical. This is the structure of the phenolic compound, which contains 2 hydroxyl group, and this is the free radical. In the first step, the free radical remove this hydrogen atoms from the antioxidant, and that become itself free radical. And the second mechanism, we can see the antioxidant, this molecule, can give this electron to the free radical and becoming itself free radical. In the end of these steps, we can see the 2 molecules are stable. So this is the mechanism how the antioxidant inhibits the free radical. This is the step for the quantitative total phenolic content and the antioxidant for the chili wast. The sample is drying. Then after drying, we separated the seeds from the tissue and grinding, then make the extraction. The extraction involves many steps like filtration, centrifuge, and evaporation, then filtration again. The extract, when we get the extract, we make the analysis for the total phenolic compound and anti-activity and antioxidant activity. We got our sample from a local manufacturer, which released hundreds tons of the chili waste in a year. The sample is contain moisture. The moisture in the sample about 80 to 85%. So we stored the sample in -20 Celsius. When we make the extraction, we dry the sample and freeze drying. It takes from 72 to 80 hours, 72 hours for drying. After drying, we separate the seeds from the tissue. So I'm working in the 3 samples: seeds, tissue, and mixture. And for this 3 samples, I make the extraction, then quantification, then determine the antioxidant activity for each sample. For the extraction, this is the summarize the steps for the extraction. We follow alarm procedure with some modification. We use the shaker, we use the evaporator, and we make the extraction 2 times in each sample. We use the methanol 80% as a solvent. For the identification, the phenolic compound, there is many protocols that analysis the method for the identification. As for example, spectrophotometry, gas chromatography, thin layer chromatography, HPLC chromatography, and capillary electrophoresis, and near intraocular spectroscopy. In my research, I follow the spectrophotometry, which is colorimetric method, measure the reflection properties of the materials as a function of the wavelength. The advantage of this method is less time consumption. Also, it's inexpensive and is reproducible. Here is this is the procedure we follow. The procedure for determining a total phenolic compound Pauline method, which is depend on reduction based on reduction of the complex biphenols. And for the total phenolic, for the antioxidant activity, we followed the DPPH assay. And this for the DPPH assay is a common spectrophotometric oxidant. That advantage of this method is that the antioxidant is measured in the ambient temperature. So the degradation of the molecular tested is eliminated. For the results, we use for the result we have to prepare the standard curve of the gallic acid. This standard curve helps us to obtain the concentration of the phenolic compound. So the result of were expressed as the gallic acid equivalent per gram of the dry sample. So we used this this graph to obtain the corresponding concentration for the absorption, depending on this equation. This is the result that I got from the three samples. I have seeds. This is S, it's referred to seeds, and P is referred to the skin, and M refers to the mixture of seeds and the skin. We can see here the concentration, the concentration of the mean average concentration of the three samples. Each sample is analyzed in a triplicant. And here is the final concentration of the phenolic compound in each sample. For we can see here from the seeds, we got 10, the concentration. For the pill, about 16. For the mixture is between them about 14. For the antioxidant activity, for the seeds, we got 20, about 20, and the skin about 12. For the mixture is between them. From this graph, we can see here, in this is the antioxidant activity for the seed is significant, the higher antioxidant activity in the seeds, followed by the mixture, then the tissue. For the antioxidant, for the phenol compound, we can see the tissue has the higher evidence of the total phenol compound, followed by the mixture, then the seeds. For the conclusion, the phenolics content and antioxidant activity for the chili waste was assessed. The content of the phenolics were higher in skin than seeds, and that can be attributed to the reducing sugar in the tissue. And for this point, I'm gonna make the analysis for the reducing sugar in the tissue to comparing that with my results. The antioxidant activity were higher in the seed than in the skin, which is expected because the seeds contain a higher flavonoids, which is a higher antioxidant activity. And the bioactive compound recovery from the processing industries not only add value to the food waste, but also be useful in reducing cost of the formulated products and decreasing the use of synthetic chemical in such formulation. As a future work, this study, the result will aid determining the market value of the ingredients of functional foods. Thank you for everyone. And if you have any questions.
Questions for Asmaa?
Yes.
Thank you, Asmaa, for your very interesting talk. Okay, Asmaa, what kind of phenolics do you have in Chile?
Actually, we until now we can determine the exactly what the kind of phenolic in the chili, but we determine the amount, the concentration, and we determine the antioxidant activity. But depending on the literatures, the flavonoids is the higher concentration in the chili.
Flavonoids?
Yeah.
Okay. So do you think you have an influence on the drying of the chili waste on the concentration of phenolics? Because some of the flavonoids are sensitive to temperature.
Yes, but we use the, yes, exactly. But we use the freeze drying for the drying, which is the temperature cannot influence the result in the freeze drying.
We do have a question in the chat from Govinda. What ratio of seed and peel was there in the mixture?
Excuse me. Let me see the, oh, sorry. Can you repeat, please?
The question by Govinda is, what ratio of seed and peel was there in the mixture?
And the mixture is 14. You mean the final concentration?
I guess I'm just reading the questions.
Okay, Asmaa, let me put this question.
Yeah, please.
In some other way. I asked this question that what ratio of seed and peel was there in the mixture? It is because here in DPP as antioxidant activity, you are showing that seed has the highest, followed by mixture and the peel. So did you use the same ratio of seed and peel for the mixture? Otherwise, there might not be space for comparison, because you are, if the ratio is different, then probably there might be some questions regarding that. So what was the ratio of seed and peel in the mixture?
The mixture is, when we take the mixture, we take it as randomly.
Okay, so you didn't actually measure the how much amount of seed and peel was there in the mixture,
no.
right? It was
no, Governor, because we took the mixture randomly.
Okay, all right.
Yeah.
Thank you.
Yeah.
Well, we want to thank you, Asmaa, for your talk, and let me give the floor to Sergio